Okay, so the next thing we're going to talk about is Mendel and his contributions. The first one is I can explain his contributions to the field of genetics. He had 4 main contributions. So he was the one that studied the pea plants. And as he came to the ends of his experiment and was forming his conclusions, he came up with a couple things. There are different variations of a gene. And we call these alleles. He didn't call those alleles, we did. And he said there were only 2 because the plants that he was working with, there were only 2 at the time. Number 2, sorry, there's fireworks going on outside. For every gene that you get, and he didn't call it a gene, they weren't called genes at this time, he just called them characteristics, inherited characteristics. There are 2 alleles that you will have for a gene, one from mom and one from dad. Okay. He also came up with the principle of dominance, which simply stated that for all these alleles, for all these genes, these inherited characteristics he was studying, there were 2 versions, and one was dominant over the other. What that meant was if the dominant one was present, that physical trait would show up. If the recessive was present, it would only show up if both of the alleles were recessive. If you have a dominant there, it's going to mask that. And it just so happened that the 7 genes that he worked with, the 7 inherited characteristics that he worked with, were all simple dominants. They were 2 alleles, one was completely dominant over the other, and so that was why he was able to come to these conclusions. And then the 4th thing is the principle or law of segregation. What that talks about is if you think back to meiosis, when chromosomes are going through meiosis, they're being separated out into those 4 daughter cells through the processes of meiosis 1 and 2, and that separation and segregation mean the same thing. And so when you separate those chromosomes out into those 4 daughter cells, that is segregating them. And so he figured that out. Even though he couldn't see chromosomes, even though he didn't know what a gene was, he was able to figure out that those inherited characteristics sorted out into different sperm and different eggs. So those were his 4 main contributions. I can define allele. Allele is just a variation of a gene. So you have, let's say, the gene for flower color, and it might be purple and it might be white. And so those would be the 2 alleles. And what makes them different is the sequence. And it may be 1 letter difference. It may be like, AAATTT and then AAATTC, and that changes the color from purple to white. I mean, it could be something that small, or it could be a larger difference in the sequence. I can describe what it means for an allele to be dominant. Dominant alleles will show up if they're present. It doesn't matter if you have 1 copy or 2. If they're there, you will see that trait, and that's what it means to be dominant. They mask the recessive trait. So even in a heterozygous organism, where they are like big T, little t, it's like that little t is not even there because it doesn't get to speak up. If this is tall and short, this plant would be tall. So the short gene doesn't even matter. I can describe what it means for an allele to be recessive. Recessive alleles will always be masked by dominant, so the only time you will see a recessive trait is when you have 2 recessive alleles. So again, if you have a tall, the plant's going to be tall. The only way the plant can be short is if it inherits a short gene from mom and a short gene from dad. I can explain the law of segregation. That's just saying that chromosomes will separate, and I didn't draw mom in red like I've been doing, but as this plant or the animal or the whatever goes through meiosis, those chromosomes will separate out into those 4 daughter cells. I can define genotype. Genotype is what your genes say. We show this with big T, little t. In reality, it is a sequence of DNA that gives you that codes for a specific protein. Like I was talking about, the AAATTT might give you purple and the AAATTC might give you white. But what we show that with is we show that with letters. And so dominant letters are shown or dominant genes are shown with a capital letter. Recessive are shown with a lowercase. But that's the genotype. Phenotype is physical. So just remember, phenotype starts with PH, and so does physical. And so the phenotype is the physical result of that genotype. And so the phenotype with this would be tall, and the phenotype for this plant would be short. Now, it doesn't mean I can always look at you and see your phenotype. So something like your blood type would also be a phenotype. So if you have type A blood, that's a phenotype because that is something we can physically see. We have to test for it, but we can physically see it, but I can't see it when I look at you. So don't think that phenotypes are necessarily always something you can see by looking at somebody, but it's just the physical expression of that gene. And that's that. So that's a little bit about Mendel and how he contributed to the field of genetics and a couple of terms that we use as our foundation for genetics. And I hope that helps.